Hi guys, Miss Francis here. I would like to talk to you today about facilitated diffusion and the sodium-potassium pump, which is a form of active transport. We're going to start with facilitated diffusion, and what I did this time was I actually showed this animation before we took notes, and that seemed to help people grasp the concept. I find that if you see it and then take notes, sometimes it's easier to understand. So let's dive in. Well, facilitative diffusion involves the use of a membrane protein because some molecules need a little help getting across the membrane. For example, molecules that are polar or molecules that are ionic, that are charged, are going to need some help passing through both the hydrophilic and hydrophobic areas of the cell membrane. I also, this section of notes, tried to model how you might want to take your notes. I've noticed that you guys are trying to copy down every single word, which isn't an effective way to take notes. Notes should be summaries of what you're seeing on the screen. I realize the PowerPoints have lots and lots of words, so I'm going to model with you guys how you might want to take your notes. For example, what can we say about facilitated diffusion? Number one, it's a type of passive transport. Number two, it involves moving from high concentration to low concentration and therefore requires no ATP. Well, which type of molecules require this facilitated diffusion? Polar molecules and charged molecules such as ions. For example, water uses the membrane protein known as aquaporin. Glucose uses the membrane protein known as GLUT. And you guys read about aquaporin in your homework one night. Well, how are these transport proteins like enzymes? And you guys actually have this slide regarding the similarities between transport proteins and enzymes. So what can you write in your notes? Well, you could say that transport proteins have specific binding sites like enzymes. They can be used more than once like enzymes, and they can also become saturated similar to enzymes. The next area we discussed was channel proteins, which are another type of membrane proteins. Channel proteins are, for example, your aquaporin. So what can we say about channel proteins? Well, they act like corridors. They act like tunnels, and they're not going to require any energy. Next, we discussed another type of membrane protein known as gated channels. Gated channels is just another word for a gated channel protein. Now, this type of membrane protein works a little bit differently than a channel protein because it requires a chemical stimulus. That chemical stimulus transforms the shape of the molecule. It sometimes opens up what's known as a gate, which then enables the molecule to be transported across the membrane. So here's a little information about the gated channels, right? It requires a chemical stimulus. One example that we're going to get to are neurotransmitters, which are going to allow sodium ions into the cell. This little simulation right here compares and contrasts gated and non-gated channel proteins. But now, some molecules need to move from an area where there's already a high concentration of them. Since we're moving from a low concentration to a high concentration, I'm going against the concentration gradient. So now I need the use of energy. I need to use ATP. And this type of transport is critical for a cell to maintain its internal concentrations of small molecules that would otherwise diffuse across the membrane. The natural inclination is for something to move from a high concentration to a low concentration. Well, let's say, for example, I already have a lot of glucose inside my cell, but I want more glucose inside my cell so I can store it and use it as an energy reserve. I might use active transport to get that glucose into my cell. So how does ATP power active transport? What happens is ATP loses a phosphate. That bond that was holding on that third phosphate in adenosine triphosphate contains a lot of energy. That energy that was stored in the bond is what enables the transport protein to move the solute across the membrane. So what can we write in our notes about active transport? Well, molecules are moving from a low to a high concentration. It requires energy, known as ATP, and this is important because it's going to maintain the proper balance of small molecules. The sodium-potassium pump is a form of active transport. It's a pump that uses active transport in order to get sodium ions and potassium ions across the membrane. So how does this sodium-potassium pump work? Well, let's go through it. First of all, a couple of things to point out in this diagram. And before we even got to the diagram, I showed these two videos which kind of help you conceptualize what's going on. What I want to point out is that this is the extracellular fluid and this is the cytoplasm. In other words, this is the plasma membrane that's around this cell. Where inside the cell, I have a high potassium concentration and a low sodium concentration, and outside of the cell, I have a high sodium concentration and a low potassium concentration. Well, where are those three sodiums going to be pumped? They're going to be pumped into the extracellular fluid. Sorry, I spelled that wrong. And where are the potassium going to be pumped? They're actually going to go from a low to a high concentration. They're going from the extracellular fluid to the cytoplasm. For each exchange to occur, it requires one ATP. So what's the process? Well, first of all, sodium, 3 molecules of sodium, are going to bind to our protein that is embedded in the membrane while it has the configuration of it looking like it's open towards the cytoplasm. And this occurs once ATP is phosphorylated. Once that happens, it causes a conformational change. In other words, now my protein opens to the extracellular fluid and my sodium, my 3 sodium, are released. In this new conformation, the potassium have an affinity towards this membrane protein, and those potassium will bind to the membrane protein, releasing the phosphate group. That triggers another conformational change. So now my membrane protein goes back to its original conformation where it's open towards the cytoplasm. The potassium is released and the sodium sites are now receptive again. They have, there's an affinity for sodium to bind again. Now notice the ratio of sodium to potassium. So I'm pumping 3 positive molecules out, but only 2 molecules back in. And what this does is set up a gradient where I have a higher positive concentration on the outside and a lower positive concentration on the inside. That's what we're going to talk about next time. Let's talk to you then.